heme part or the iron part. So here it has been written the RBCs of the human contain red colored pigment having iron called hemoglobin. I told you hemoglobin is made up of mainly two components. One is iron and another is globulin protein. So here iron is also called as heme. So here heme is giving the red color to the RBCs. For RBCs are one of the main component of the whole blood cell. So here RBCs are in red color just because of this hemoglobin, and especially by this iron part, our RBCs look red in color. So here RBCs of the human contain a red color pigment having iron called hemoglobin. It binds with oxygen in a reversible reaction manner. Reversible reaction manner means what? The heme part or the iron part of the hemoglobin sometimes binds with oxygen and sometimes it will release. Means reversible reaction manner means the sometimes it will bind and sometimes it will release. Okay, both the things will happen with this, with the binding or affinity of this hemoglobin with the oxygen. A single hemoglobin, this line is so much important as far as your exam is concerned. A single hemoglobin molecule can bind with four oxygen molecule. This question has been asked several times in different examinations. Even though in competitive examinations too, this question has been asked that the single hemoglobin means one hemoglobin binds with how many molecules of oxygen. So you can see here that a single hemoglobin molecule binds with four oxygen molecules. You will have to remember this data. Okay, now the binding capacity of the oxygen depends on the partial pressure of oxygen. Mainly I told you na ki there are certain conditions in which the hemoglobin will bind the oxygen, and there will be the different condition in which the same hemoglobin which binds the oxygen will release the oxygen, and this is called the reversible reaction. Okay, so here it has been written binding capacity. Here the capacity of the binding of this, or capacity of binding, or we can say the affinity of the hemoglobin towards the oxygen mainly depends on partial pressure of oxygen. Means wherever the partial pressure of the oxygen is high, then it will promote the binding of oxygen with the hemoglobin. Okay, but other factors such as PCOT, partial pressure of carbon dioxide, hydrogen ion concentration and temperature, etc., also regulates its binding. But mainly one factor plays an important role, that is the partial pressure of oxygen. Okay, po2, if the po2 will be high, and rest of then what will happen, it will promote the binding of oxygen with the hemoglobin, first thing. And the second important thing you will have to remember, that not only the partial pressure of oxygen is there, some more factors are also there. Some more factors are also associated with this binding or the affinity of this oxygen with this hemoglobin. What are those factors? Partial pressure of carbon dioxide also, hydrogen ion concentration also, and temperature also. Means three more factors are also there which will promote the binding of this oxygen with the hemoglobin. Okay, not only this one, not only partial pressure of oxygen, but mainly it regulates the binding. Okay, now we'll see this PCO2 means partial pressure of carbon dioxide, hydrogen ion concentration, temperature, etc., interferes with this binding. So what happens? Although the partial pressure of oxygen promotes the binding, but partial pressure of carbon dioxide, hydrogen ion concentration, and the temperature, all these three factors will also regulate its binding. And what they do, they interferes with this binding. So if the partial pressure of the oxygen will be high, then it will promote the binding, and in certain conditions the PCO2, hydrogen ion concentration, temperature, all these three factors also play an important role sometimes, as they interferes when their concentration becomes different, then they interfere with the binding. Okay, while plotting the graph between the partial pressure of oxygen and percentage saturation, we obtain a sigmoid curve. Okay, you just see this one is the sigmoid curve, which is called as oxygen dissociation curve. So sometimes in certain examinations this question has been asked, what kind of curve we can get when we are plotting the graph between the partial pressure of oxygen and percentage saturation. So what this graph is depicting, you could see here, this is a sigmoid curve. So this scale x-axis is representing nothing but just the partial pressure of oxygen, and here this axis is representing the percentage saturation of the hemoglobin with the oxygen. So what happens when we are, you just see this one, this scale where where we are representing the partial pressure of oxygen. Whenever we are increasing the partial pressure of oxygen, then the saturation, percentage saturation increases at a higher rate. You just see at a higher rate it is increasing, and after some time it becomes linear. But after some times what happens, the saturation, after some time the graph becomes plateau. Means in the last part of this sigmoid curve, you could see that due to, although we are increasing the partial pressure of oxygen, but the hemoglobin will become the saturated, because the number of hemoglobin is limited, because the number of RBCs are limited. So if you are if you are increasing the partial pressure of oxygen, then in first stage it will, saturation will, the rate of saturation will run at higher rate, then it becomes linear, and after some time it will be saturated, or we can say it will become plateau. Okay, it means after this point, if you are increasing the concentration of oxygen and if you are increasing the partial pressure of oxygen, then what will happen, the whatever hemoglobin is there, those will be saturated, and after that no binding of oxygen takes place. Means up to a certain time, up to a certain concentration, up to a certain partial pressure of oxygen, hemoglobin allows the binding of oxygen, but after a certain limit it will not allow, because you know that only four more oxygen molecules can bind with a single hemoglobin molecule, and the number of hemoglobin molecule is limited in the human body. Okay, so due to limitation of the number of the number, or we can say the amount of hemoglobin molecule in the human body, after some time of saturation we can see, and this curve, this sigmoid curve is called oxygen dissociation curve. Okay, now we'll see the different conditions in different places, and in these different conditions sometimes this hemoglobin allows the binding of oxygen. You just see in alveoli, you know that blood captures, or we can say the hemoglobin captures or binds oxygen, and it transports the oxygen, and it transports it catches the oxygen or binds the oxygen, and it reaches to the different tissues or the cells and releases it. Okay, so here you just see hemoglobin, H, capital H and B is generally, we are using the term to represent the hemoglobin. So here you just see that this hemoglobin binds the oxygen in alveoli. But the same hemoglobin which has captured the oxygen, or you just see this hemoglobin has oxygen, and in tissues the same hemoglobin is releasing the oxygen. Means in some condition, you just see there will be the I told you that the condition only regulates the tissue condition or the cells condition only regulates the binding and unbinding of oxygen with the hemoglobin. Okay, so here you know that in alveoli this hemoglobin has to capture the oxygen so that it can transport the oxygen, because around 97% of the oxygen is transported by hemoglobin. So this binding is so much necessary, and this necessary reaction, this binding takes place in alveoli in certain conditions. You just see what are those conditions. High O2. Okay, I told you the main or important role is played by the partial pressure of oxygen. So in alveoli, this condition is there which helps the oxyhemoglobin formation. This one will be called as oxyhemoglobin. Means when hemoglobin binds with oxygen, then this hemoglobin will be called as oxyhemoglobin. Means what we can say, this condition will promote the binding of oxygen with this hemoglobin. So what would be the condition? Here you could see that in case of alveoli, you know the alveoli is the main place where where the exchange of gases takes place, where the blood cells receive oxygen, where the blood is releasing carbon dioxide. So this is the place where gaseous exchange takes place. You will have to remember this point. Alveoli is the place in the lung. Okay, this one is the terminal part of the lung. Okay, so here you just see that high oxygen or partial pressure of oxygen, low CO2, less H plus means less hydrogen ion concentration, and low temperature. So all these conditions promote the binding of oxygen to hemoglobin. Means obviously in case of alveoli, when we are inhaling the air, then the concentration of the oxygen should be high. Okay, and obviously it is a high. So the partial pressure of oxygen in inhaled air is high. Due to this, the amount of oxygen is high. First thing. The second thing, in environmental air as compared to the oxygen, the amount of carbon dioxide is low. So here you just see low carbon dioxide. I told you not only partial pressure of oxygen is regulating its binding, three more important things are also there. You just see here the amount of carbon dioxide, the number of H plus ion, and the temperature. So here low temperature, less hydrogen ion concentration, low PuCO2 or low CO2, or high PO2 or high O2. So when the amount of oxygen is high, amount of carbon dioxide is low, the number of H plus hydrogen ion is less, and the temperature is less, then this condition promotes the binding of oxygen with the hemoglobin, and here hemoglobin becomes oxyhemoglobin. First thing, on first side we are talking about. Here on second side, so here you just see hemoglobin binds the oxygen and it transports the oxygen to different tissues or organ, or we can say ultimately to the cell. Okay, so the different condition in the cell will be there, which will promote the releasing of this oxygen. You just see here dissociation of this oxygen takes place with the hemoglobin. Okay, dissociation of oxygen takes place in this with this hemoglobin, and here you just see hemoglobin. Previously this hemoglobin binds this oxygen, now this the same hemoglobin is releasing the oxygen. Okay, now we'll see what would be the conditions. So there would be certain different conditions which will promote the dissociation of oxygen with the oxyhemoglobin. So now the oxyhemoglobin becomes the hemoglobin only. Okay, now what would be the different condition? You could see here low O2. You just see previously for binding, the partial pressure of oxygen should be high, but here the partial pressure of oxygen would be less, partial pressure of carbon dioxide will be high. Here the hydrogen ion concentration, or we can say the number of hydrogen ion, will be more, and here the temperature will be high. So you just see just reverse condition is present in the tissue, and due to this reverse condition, due to the deviated condition, what will happen? The whole of these four factors promote the dissociation of oxygen with the oxyhemoglobin, and here oxygen will be released. So you can see the overall perspective of this binding and unbinding. You just see when hemoglobin or the when RBC reaches to the alveoli with the hemoglobin, there are certain conditions through which the hemoglobin binds oxygen, and after this binding it becomes oxyhemoglobin, and this oxyhemoglobin moves from the lung or from the alveoli to different tissues, and in different tissues the conditions becomes different. You just see these conditions will be there, and what would be the different condition? Here the partial pressure of the oxygen is low, and how you can remember that the oxygen partial pressure of oxygen is low? Just because the tissue is consuming all the oxygen, because all the time this tissue or the cell is consuming oxygen in order to make the ATP. So all the time this cell or the tissue needs energy. So all the time what transported either by any of the way, it will consume it, and after consumption obviously the concentration becomes low. And you know that after oxidation of the food, during the formation of ATP, carbon dioxide is produced. It means obviously the partial pressure of carbon dioxide, or we can say the amount of carbon dioxide will be high in tissue. So you just see here and here, just reverse of this. Here the previously here the hydrogen and concentration was less, and here the hydrogen and concentration is more, and here in case of alveoli the temperature was low, and here the temperature is so much high. Okay, so in this condition the hemoglobin is releasing oxygen. So by this way the blood is capturing the oxygen in lungs, or we can say the alveoli, and the oxygen is the RBC is transporting oxygen, and it reaches to the tissue and releasing the oxygen. So this is the phenomena. Okay, so by this way the oxygen is being transported, and 97% of the oxygen is being transported, and I told you 3% of the oxygen is transported in dissolved form. So obviously in dissolved form oxygen will be transported from one region to another, and due to concentration gradient oxygen will be diffused from one region to another. I told you that how the concentration gradients will be created in different sites. Okay, and due to the difference in their concentration the oxygen and carbon dioxide will be transported. Okay, now two important facts are there, you will have to remember those. First fact, 100 ml of the blood carry 5 ml of oxygen. Okay, so you will have to remember around 100 ml of the blood is generally carrying 5 ml of the oxygen, and the same amount of blood means 100 ml blood is carrying 4 ml of carbon dioxide. So you will have to remember 100 ml of the blood is carrying 5 ml of the oxygen, and 100 ml of the blood is carrying 4 ml of the oxygen. So you will have to remember this data. Now we'll come on the important aspect of this chapter, very much important aspect of this chapter, transportation of carbon dioxide. In the beginning only I told you this one is so much complicated, and based on this complicated one several of the questions are being asked in several of the competitive examinations too. Okay, so here we will see how the carbon dioxide will be transported. So I told you that carbon dioxide is being transported by three ways. One, I told you that it is being transported by hemoglobin by RBC, and the second I told you that it is transported in the form of carbonic acid or carbonate ion, and the third I told you that 7% of the carbon dioxide is transported in dissolved form in the plasma. Okay, so one by one we will see how the hemoglobin is transporting the oxygen, how the means how this carbon dioxide is being transported in different organs in different way, in different conditions, how the same carbon dioxide is binding with the hemoglobin, and the same hemoglobin is releasing the same carbon dioxide. So we will see one by one here all these three types or the ways of the transportation of carbon dioxide. Now we'll see now we'll first discuss first of this heading here in the form of carbaminohemoglobin. So I told you when hemoglobin binds with oxygen, then it will be called as oxyhemoglobin. And here, when the same hemoglobin binds with carbon dioxide, then it will be called as carbaminohemoglobin. Means when hemoglobin, I told you that 20 to 25% of the carbon dioxide is transported by hemoglobin. And how it transports the carbon dioxide? It binds with the carbon dioxide and it transports it. So when the hemoglobin is binding with oxygen, then it will be called as oxyhemoglobin. When the same hemoglobin binds with the carbon dioxide, then this hemoglobin or the binded form of the hemoglobin with the carbon dioxide will be called as carbaminohemoglobin. So you will have to remember that this point. Okay, so we will see here one by one each of the line. Carbon dioxide transports through hemoglobin in the form of carbaminohemoglobin around 20 to 25%. I told you before. HbCO2. Now it will be called as means in short form you can write here HbCO2. Hb means hemoglobin, and carbon dioxide is present with this hemoglobin in binded form, which solely depends on PCO2 and PO2. Okay, so I told you certain conditions, certain conditions would be there, which will promote the binding of carbon dioxide with the hemoglobin and unbinding of the carbon dioxide with the hemoglobin. So you just see here the binding and unbinding of this carbon dioxide with this hemoglobin depends on partial pressure of carbon dioxide and partial pressure of oxygen. Okay, so here two things are there. Previously we have seen the binding of oxygen depends on four factors, but when we are talking about the binding of carbon dioxide with the hemoglobin, only two factors are involved here: temperature and temperature, as well as the hydrogen ion concentration is not playing a specific role in this particular process. Okay, so here which solely depends on partial pressure of carbon dioxide and partial pressure of oxygen. In tissue, PCO2 is high, obviously, since tissue is releasing a lot of carbon dioxide by consuming a lot of oxygen. So here in tissue, PCO2 is high and PO2 is low. Obviously, this would be the condition in the tissue. So there will be more binding of carbon dioxide takes place with the hemoglobin. So what happens in tissue? What will happen? The amount of carbon dioxide will be high and amount of oxygen will be less. And this condition promotes the binding of hemoglobin with carbon dioxide. However, in lungs, the just opposite condition would be there. So here what happens? You know that in case of lungs, PCO2 is less and PO2 is high. Okay, which leads to dissociation of carbon dioxide from carbaminohemoglobin. So you just see both the conditions in this depicting picture. You could clearly compare both the tissues and or both the condition in which both the binding and unbinding of the carbon dioxide is taking place. Here you could see in tissue the amount of carbon dioxide is high and amount of oxygen is low, and due to this concentration, due to high concentration of carbon dioxide and low concentration of oxygen, what will happen? When the amount of carbon dioxide increases, then it promotes the binding of carbon dioxide with hemoglobin, and here you will see hemoglobin binds with carbon dioxide and it forms finally carbaminohemoglobin. Okay, and after binding the carbon dioxide, the hemoglobin moves from the tissue to the lungs, because this carbon dioxide has to be expelled out from the lungs. So this carbon dioxide which has been formed in the tissue, and this there is no role of this carbon dioxide, and there is no essentiality, there is no need of this carbon dioxide, or we can say this carbon dioxide is a waste product of the oxygen, you know? So this carbon dioxide has to be expelled out from the body. So hemoglobin catches it, captures it, or binds with the carbon dioxide. It becomes, the hemoglobin becomes a carbaminohemoglobin, and blood, or we can say the RBC, or we can say the hemoglobin of the RBC of the blood transports this carbon dioxide, and it reaches finally into the lung, and where it reaches to the actually in the alveoli part of the lung, then this carbaminohemoglobin dissociates, and when this carbaminohemoglobin dissociates, then it releases carbon dioxide, and you just see when it releases, means the hemoglobin releases carbon dioxide, then this carbon dioxide becomes free in the alveoli. And how the same hemoglobin binds with the carbon dioxide here, and the same hemoglobin releasing this carbon dioxide in this particular place, just because of this condition. You just see here partial pressure of carbon dioxide is low and partial pressure of oxygen is high. So we can say that in case of alveoli of the lung, the concentration of carbon dioxide is low and concentration of oxygen is so much high. This promotes the unbinding of carbon dioxide with the hemoglobin, and it promotes the dissociation of carbon dioxide, and by this way the carbon dioxide will be released and from this hemoglobin. And when the carbon dioxide will dissociate, when the bond which was formed before with this hemoglobin will be broken, then carbon dioxide becomes free, and here when it becomes free, then it will be expelled by the process of expiration. So you know that 20 to 25% of the carbon dioxide is transported by both of these ways. Means here the binding of carbon dioxide takes place, and here the release of carbon dioxide takes place.